Understanding Gene Cloning, a simple guide. Hello everyone. Today, we're going to explore the fascinating world of gene cloning. This concept is not just a staple of science fiction, but a real scientific practice with incredible implications. Whether you're a student, a curious learner, or someone interested in the marvels of genetics, this video will help you understand what gene cloning is all about. Let's dive in. Gene cloning is a process where scientists create exact copies, or clones, of a specific gene. This is achieved by isolating and copying the DNA sequence of interest. The key here is the DNA, the molecule that carries genetic information in living organisms. By copying a gene, scientists can study its functions, produce proteins for treatments, and even modify organisms for desired traits. 1. Isolation of the gene. The first step is to identify and isolate the gene that needs to be cloned. 2. Insertion into a vector. The isolated gene is then inserted into a DNA molecule known as a vector. This vector acts as a carrier or a delivery system. 3. Transformation. The vector with the new gene is introduced into a host cell, often a bacterium. 4. Replication and cloning. The host cell replicates, creating copies of the gene as it divides. 5. Identification and harvesting. Finally, scientists identify cells that have successfully cloned the gene and use them for further study or application. Gene cloning has a wide range of applications. Medical research, producing proteins for treatments, like insulin for diabetes. Genetic engineering, modifying plants for better yield or resistance to pests. Research, understanding genetic diseases and developing gene therapies. We hope this video helped you understand the concept of gene cloning. It's a tool that holds immense potential in various fields, from medicine to agriculture. Remember, gene cloning is about copying and studying specific genes to unlock the mysteries of life. Thanks for watching, and stay curious.